is visible to
Yeah,
us.
yeah. So, uh, sir, okay. Should I start, sir?
Yeah, definitely, sir.
Thank you, sir. Thank you. Okay. Okay. I think this one is not hiding. Okay, we'll continue with this only. Okay, I will continue with this only. It is not going any.
What happened, sir?
I think you are seeing something on screen.
Yeah,
That is this particular
deep neural
screenshot.
networking.
Yeah. Okay, sir. Okay, we'll go with this. Okay,
Yeah,
perfect. Yeah,
yeah.
thanks. So we were just dealing with the type of neural network system, and we were dealing with the two type of neural network system, such as deep neural network system, and second one is a recruiting neural network system. These both the systems are widely used in drug design right now. So the deep neural network system is just similar to the neurons which are available in our brain, which are being quite related to the artificial intelligence. So these neurons are being connected to each other, and the input layer and output layer is connected with the multiple hidden layers. These hidden layers are basically required for the processing of all the input data what it will get. It will process here and convert it into the results. So the incoming signals, whatever the signals are coming with the input layer, this particular hidden layer consists of number of weights. It may be whatever be the weights we are allotting to them. We are giving the weights to these hidden systems, and they are multiplying with the hidden systems, and that weights are multiplied and converted into the results which are come into the outermost and out layer. The output layer will come as a signal Zj, and that the sum of this all the product will be like at Zj is the summation of the W1 weight into the A1 plus B1. So this indicate this totally indicates the calculation of artificially learning and calculation of the particular results from deep neural learnings. The same thing quite in a different way occur in a recruitment neural network system. The recruitment network system is of two type, and the two type include RNN and UGRNN. The RNN is just calculation of a sequential data, and that sequential data here is indicate in the form of H1. Here this H1 data basically depends on the previous data's element output report, and the hence it depends on the previous element output result. It means it consists of memory. So this RNN system is basically learning with memory system. It basically based on memory of the previous neuron. So once one neuron will give the result, that will be carry forward with the second neuron. So this in hidden system goes with the sequential data only. So by calculating this, we can easily evaluate the results. The same thing occurs while in a graphical unidirected graph form with the use of UGRNN system. In UGRNN system, they basically depends on the root nodes. What we will do is, suppose this is an example of a particular structure consists of amide group. So what we will do is, if we have two carbons, if we will calculate, there are three carbons, but the two methyl groups are being attached with the amide group, amide functional group. One is at N nitrogen, and one is with the amide group RCONH2. So here this carbon, there are three carbons now, 1, 2, and 3. These three carbons are being considered as root 1, and the nitrogen and oxygen are considered as another root. So total number of root will become 5. So you can see with this is a carbon number which is considered as root 1, nitrogen root 2, this this oxygen root 2, this nitrogen root 3, another carbon is root 4, and the final carbon is known as root 5. These are the considered as the roots, and according to this root we will calculate the with the help of neural network system, the results. So now, when we compared it with the traditional drug discovery method, we will find that whenever we will go with the traditional drug discovery method, we required a drug targetable, as it should have a well-defined target, and then we will go for the interaction study. However, there is a limitations in the traditional drug discovery method, that we do not know about the actual cellular interaction, and that is the limitation of the computer-aided drug design. Now, this limitation has been overcome with the use of artificial intelligence. Then artificial intelligence can interfere with that, and with the use of complex algorithm and machine learning, we'll get the meaningful information of large data, and that to be connected with the cellular condition. So if we'll take an example of RNA sequencing, we can easily identify the gene expression that is correlated with the cellular condition. So whatever be the internal data which are being not procured with the help of computer aided design, the artificial intelligence had help in that. The second most important help which we will get with the artificial intelligence is when we go for a computer aided drug design, we need a drug targets and a targetable targets. But in the case of artificial intelligence, the undruggatable targets can also be the undruggatable targets can also be identified, and that can also be used for the evolution process, and can be used for the estimation of drug interaction with the use of or we can say within a limited period of time. So this is one of the biggest help of artificial intelligence. We can see with the comparatively differentiation between the traditional and AI based. So traditional is a target based or target driven, on why when we go for a AI based, it is a data driven. And we'll find that the drug targetable, it should be a well defined target, but we won't get the cellular interaction studies with the traditional data. However, in AI based, it is a complex algorithm or machine learning which give the exact meaning information of that particular largest data, whatever be binded with the cellular interaction. So there are limitation of that complex nature identification in traditional method. However, in the AI based, we can go with the undruggatable targets which are being not been identified, so that we can easily go with this particular AI based. So there are a lot of advantages of AI based drug design over the traditional one. So this we can easily see with the one reported paper. It clearly indicates that the traditional when we go with the traditional AI approach, when we go with the comparative of traditional with the AI approach method, we will find that the heat rate is observed at 10 to 30 times as compared to a traditional experiment where heat rate is 0.01, when we observe for a virtual docking as compared to AI intelligence cursing. So this has increased. We can directly correlate it with that this can increase 100 to 300 times. The virtual docking process has been improved with the use of AI assistance. Simultaneously, if you go with reverse docking also, reverse docking, and you can easily see that in the virtual docking, we have seen that there are multiple ligands, and that is being targeted to the particular receptor. So that is a virtual docking protocol, while we will go for reverse docking where number of ligands are there, and the structures are being limited to one. And that particular thing is when we dock with the use of AI intelligence, we get the hit rate of 50 to 70%, and that is a remarkable achievement as compared to the traditional method. So hence the AI method is having a lot beneficial while using over the virtual normal common computer assisted method. So in the artificial intelligence, it basically do not rely on the predetermined target. It generally based on the learning protocol, whatever be implemented in that, and it will go for the that particular thing. So it is not a subject bias and existing knowledge. So this is a very important part of this particular drug discovery protocol. Now this also utilized AI also utilized the use of biology and computer. It is an IT based evolution. So basically when we talk about the IC based evolution, we will find it very good amount of potential of determining the predicting the interactive value. So it rapidly reduce the power and reduction of processing cost. That simultaneously, when we go with the higher predictive power, we will get a meaningful interaction, and that meaningful interaction will definitely help out into identifying the potential inhibitors or drug molecule for the particular drugs, whatever purpose we are going to screen for. And simultaneously, as we will find, the AI is very very helpful for converting from bench to virtual lab and vice versa, and it will reduce the manpower at the tremendous rate. But there are limitations of AI, since, as we know, this is an artificial system, an artificial word itself says it is artificial. We have a number of limitations with that. So the first important limitation is the development of algorithm. We need a prompt algorithm to identify that particular phrase. It should able to read the meaningful information and deliver the meaningful information. Both things depend on the algorithm, the first important thing. The second is, algorithm is biased because it depends on the creator. It is not at all process is objective. It is a subjective, because the person who developed the algorithm will totally develop the particular algorithm according to their own choice. So this is the biggest limitation of AI system, that algorithm is always on a biased basis. Now, the third important thing, although we know that the AI is basically having a supercomputer, using the high facilities of the computer, but even use of that will giving a more prompt result. But even to establish that particular lab, we required large volume of investment. So again, that is also a limitation of AI. So, finally, as I told you, that this is an artificial system. Whatever result we will get, we need to share it with the scientific validation. So that is a big problem with that. Now, how to design the AI, it is a very important challenge for artificial intelligence. So today I will give you the brief information how to develop the what to develop the AI predictors. So whenever you want to go with the work for artificial intelligence in computer aided design, you need to first define the problem. We need to first think over that. We need to go for discrimination or generation. It means we want to identify active or inactive molecule from the bulk data, or we need to generate a new molecule, or we need to develop a new molecule, that we need to identify. And according to that problem definition, the AI architect will be developed, or the algorithm will be developed according to that. So once we finalize that we want to go for a discrimination or generation, we need to develop a AI algorithm. So AI algorithm is a number of algorithms are already available. The best suited algorithm are SVM or RF models are there, and that particular models are widely used for the drug design purpose. So we can choose according to that whatever we feel that we will go for discrimination or generation, and according to that only we need to plan for a learning protocol also. So once we finalize the design or AI algorithm, we need a input data. Now the input data is very important. It should be of proper quality, proper quantity, so that we can able to process for that. If data is in a huge volume, we need to go for a data pre-processing. And after pre-processing data, we need to give a training set of that particular data. Once we will complete the training set, we will have a molecular representation, and that will help out to process the that particular AI method for that particular AI processing. Then after finishing all, we will go for a model training and evaluation. Once the everything will be developed, we need to go for a training algorithm. So there are number of training algorithm has been already been in available, but that is not enough. We can develop our own algorithm for developing that particular model. And there are number of easily developed algorithm which are available, and that are free. Some are open source also, and some are paid, which can be utilized for this estimation. So we need to train the algorithm. We need to go for optimizing strategies. We need to evaluate the particular mechanism, by which mechanism it is just calculating the particular things. We need to check out that what are the evolution parameters we are looking for. We want to go for area under curve, or we want to go for mean absolute error, we want to go for R square estimation. So we need to do for that. And whatever parameter we have included, we need to tune that particular parameter by hyperparameter tuning method. So like that, once you develop the model, then you need to go for evolution. So by step, I will tell you the how you will construct the model. So basically, if you will go and will plan for the developing a model for artificial intelligence, we need to have some thing which I have told you in previous slide. We need to have a problem definition, data preparation, design of AI architect, model training, evolution, and the important thing, the understanding and explanation of the result. Until and unless we don't know how to understand the result and explain to the result, we can't evaluate the, or we can't develop the model. So we need to understand in what form we need to understand the results, because the result is will be in a technical terms, and that technical terms we need to be converted into the understanding terms, so that learning process can be developed. So we need that. The second thing, we need to have a choice the machine learning method. We need to have an appropriate machine learning level. We have seen the three machine learning, that is supervised, unsupervised, and that is reinforcement learning. So we can employ the any type of learning protocol with this particular method, and every learning protocol will give a different type of result. If you want to go for a classification, we need to go for a learning supervised learning protocol. Then we need to identify the domain that is very important. We want to go for discriminative or generative. As already I have told you in the previous slide, we need to go for a differentiation, or we need to go for a generation of a new molecule. So we need to identify that particular domain, and according to that domain, we need to identify the architect. So once we will finalize that, we need to have some hyperparameter with the algorithm. So a number of descriptive task algorithms are available. SVM, RF, and ANN are very, very important, which are already being used. The RF, which is a robot forest method, which is already being highly employed for QSAR analysis. This is a very important and prompt method. So SVM is a very common method which is used for molecular docking and evolution purpose. So there are a number of already discriminative task methods. So we need to identify them. Once we finalize this thing, now the work is we need to finalize the hypothetical the problem solving method, that is how the input will come and how output will go, and what will be the relation between both. We need to finalize that particular relationship with them. So in such as, if we will go for SVN algorithm, we have certain parameters, kernel parameters, and error penalty parameters. We need to include that. So once we need to finalize, after finalizing the algorithm and the parameters, we need to go for identifying the layers, how much learning rate we required in that particular algorithm. And they depend on the algorithm, we need to select the number of neurons and layer we need to impart in that particular artificial intelligence. The totally, it depends, basically it depends on what type of how much depth of result you are, or how much type of learning you are wishing for that particular thing. So once you finalize that particular thing, now you need a set of a data, and that is very important, because you need a good representation, a quality and quantity of initial data. And that initial data, once you will procure that data, because that data will basically lead for the development of your right model. So once every initial architect has been developed, one may proceed for the training. We need, once we develop this full architect, we need to proceed for the training and the evolution of that particular model. So we need to finalize the data used for the particular model, and we need to develop the relationship, how the data will process and how the result will interpret it in that particular relationship. So the second important is the input data preparation. So we now here it is very, very important, as we have number of big bases which are being already available, and or we can say local databases are also there. And this database generally consists of a large volume of information. We need to first pick out the quality data, and that particular quality data will this is a actually very intensive and challenging work to have this much volume of data. So we need to identify which type of quality data particular to that domain which is being required. If you want to go for anti-HIV molecule, so you need to target which type of protein you want to which type of small molecule you want to identify. If you want to go for, suppose it is a high time for COVID-19, if you want to go for a identification of database for COVID-19, you need to identify, if you have to select the nucleotide basis type compound, you need to identify that particular domain of the molecule and need to identify the specific particular data according to that. So that particular is important. So the big important part is how well is the your input data is for the input data will directly influence the prediction of the your output value. So once if you collect the data, if required data is more, if you required, you need to go for pre-processing of the data. And so there is no any universal rule employed for that, how to process that thing. You need to fix out that particular input data by your own. So these are the type of we can say data which are being used in medicinal chemistry, which are promptly used in medicinal chemistry, and which can commonly be used in this particular AI tool. So there are we can say there is one input and output file. So there will be input and output, both type of data will be available. So if we will talk about input, we can input a number of sequences, that is biomolecular molecules. We can use the smile codes, we can use the interaction data, we can use the binding energy like interaction data, we can take a fixed size vectors, we can say molecular structure graph. This can be a part of inputting the data. We can input this type of data, and what we can get a binary output, we can get an integer output, as already we seen in the graph that time. We can get the real value number regression task, we can get the fixed size, we can get the single column data, or we can get the sequential data such as smile string or amino acid sequences. So we can finalize both the things, input data and output data, in what form. These are the following form which can be used in the medicinal chemistry easily for this particular in this particular cases. So now once you finalize the data, there is a very important thing which I already told, is the quality and quantity of initial data. And that is very, very important because it is a representation of. So how you can judge the important data, that data is of quality mark, how you can able to judge that? So you need to judge that. For that, you need to set a training set for that. Once you go for a training set, you need to check is the performance of the training cell is accepted, whether it is accepted or not accepted. Once it is accepted, you need to check the performance on a test set. Once it is also accepted, then you can achieve the model, reliable model. It is a reliable model, you can move with that. But once if the performance training set is not accepted, you need to change the AI architect. You need to change, whether you are using ANN system, then you need to go with the other system, RF system or SVM system. You need to change the AI system if the performance training set is not acceptable. And once you change the performance training system, again you check it for the performance. If performance is better, then it is very good, you can accept it and you can achieve the model. But if it is not accepted, now then it is a big trouble to you. You need to change the data, you need to clear the data, and you need to take the improved and good data in that case. So once if in that particular case, if your data is not being reliable, you need to reframe the AI system and data system. And if your data is okay, and if its performance is on test set is acceptable, then you can achieve the reliable model and you can proceed for that. But if it is not achieved, in that case, you need a high quality of data, and that for that purpose you need to see the acceptable cost for that, because the data procurement is a highly costly procedure. If it is a non-costly procedure, you collect more data, or if it is a costly procedure, then you need to check your training set. You need to increase the multiple tasking to purify your data, to clear or to make a more learning protocol. So that will make a retrained model. And once you make a retrained model, again you check your data on that. If it is achievable, that is very good, you can go with that. But if it is not achievable, the data is not reproducing, then you need to go for a collection of a new data, or either improving the learning model. You can't go with that particular learning algorithm, because that learning algorithm is not reproducing your results. So this is about how you can go further proceed for that. So after that, you need to check the training set performance, and whatever I have explained you in there, this is the critical, what to say, the troubleshoot. If that will come in training set, what are the prompt troubleshoot you can implement for that. Once you go for a training set performance and the training set performance is not acceptable, the first important thing is you need to tune or reconstruct your architect. Whatever architect AI model you have drawn, you need to retune that model, because that model is not accepting the training set, whatever the data you are imputing is there. If after tuning, if you have made tuning or change, make a change in architect, then also if you are not getting a improvement in your result, then you need to work on the training data, whatever data you are getting, because it consists of lot of error, or it might be possible it consists of noise or error and other inputs. So you need to balance that, and that for that purpose you need to look for the training data. Once you go with the training data, if further we will go for processing that particular data, you need to check the model identity performance with particular data which has been checked for. Then after that, if your training set is acceptable, you can go for it. There is no problem for that. If there is a gap between the training and test set performance, then you need to go for that is collecting the more data, because whatever data you have with you is not reproducing the training set data with the test set data. So the training set should be coincide with the test set data. So for that purpose, you need to either change your data or need more collection of data. For example, if you are working with the binding energy, so you have taken near about 100 molecules for evaluating the binding energy. So the learning protocol has developed that this particular functional group will give this much binding energy, and now his learning protocol has already developed this thing. Now the second the test sample, when test set is you applying, so this training set is not giving result according to that particular test set whatever you are giving. So then you need to what you need to do is you need to again train the model with the use of new data or increasing the data. So you need to go with the particular data, and data is, again I will tell you, the data is costlier part. So you need to check whether that particular data you can procure or you can't able to procure. If you can procure, you can go with that, or nor you need to change your AI algorithm for that. So if we talk about the the data should be already told, it should be high, what to say, quality data should be required, because that particular data is correlated with the biological activity data. Because in docking, we are going with correlating the data with the biological data purpose. So these data are already costlier, but we need to take the high quality data in that particular cases itself. So now after moving to this particular term, we need to specialize the neural network architect for that particular purpose. So once we do this particular thing, we need to check the hyperparameters. So hyperparameters will be employed for the particular this AI models. Once you implement the high performance, that is high parameter models, we need to check the performance again of the test set, the adjusted regulation hyperparameter, and the generalized error parameter with our algorithm itself. So once we check with that particular algorithm, we will get the definite the high quality of data. So in that particular situation, when we are not getting a sufficient amount of, or we can say sufficient amount of results, we need to look again for the data. We need to again check for the data, because if it will take the, I have given the example of binding affinity. So we will find we are getting a huge amount of data with IC50 value, but while calculating the binding affinities, we are having a very limited data with the biological efficacy data. So that we need to identify that there is a limitation of data, good quality data. For the training purpose, we need to reframe the AI model if we are not having a good training data. So even after setting all the data, we are getting a reliable result, and we can able to underline the problem itself. The machine learning is necessarily not permit that the result will be learned totally be evaluated finally. We need to go with the evaluation by experimental finally. So because that is a very clear that cat is cat, it is not a dog, because it is a artificial intelligence method. It is not and being a learning protocol by a humans. So we need to always keep this in mind, that after calculating this, we need to check or recheck the learning system again. So further, once we check the learning system, we need to go with the training set performance. So the training set performance, we have multiple biological targets and activities. We know this about. So while it means that there will be always a problem of creating a non-linear relationship between the drug receptor and the molecule while using the artificial intelligence. As we know that artificial intelligence are not fixed to the particular targets, they can identify multiple targets in the particular protein itself. So that particular thing is the biggest challenge with the artificial intelligence. So now I am giving one example with the artificial intelligence, how our software is worked with that. So there is a one software, investigational novel drug discovery. This is a very commonly used now drug discovery software. And this approach of this particular basic is to make or to identify the active molecule for the particular set of that particular ligands, and from particular set of ligands. So it was observed that the hit rate of the index, this particular blind test software, is 30%. It means out of 100 molecules, 30 active molecules have been generated with this particular software. So for this particular software, what we need is we need to have a observed activity molecule. It means there should be a data which should have a proper and observed activity. What we need to do is we need to fragment that particular chemical observed database, and that database need to be fragmented into a smaller segments, so that we can able to relate the activity of that smaller molecules. So once we relate with that, we need to go with the screening with the large database, and from the large database, whatever the molecule being coincide or mixed or identify or assemble or similar with that particular molecule, will identify by this particular method. So first we will go with the molecules. If we'll go with the data set, we'll find, suppose if we are going for X activity, suppose we want to go for evolution of anti-HIV activity, we need to take the data set accordingly, and we need to identify what are the basic functional group which are being required for the particular action at that particular receptor, or make a what are the binding important sites which are being available at the protein. We need to identify that, and with the use of this data set, we can able to identify the what are the important functional group. Once data set is being set, we need to go for a fragmentation, and that fragmentation is being used with the AI tool. So molecule breaks in a chemical relevant fragments. The simple fragments which is being break in the molecules and its components. So whatever be the ionic form of the molecule, or what are the small molecule segment or hydrophobic or hydrophilic part, they might be this type of fragments we will get from that. Once we will get the fragmented part, we can easily evaluate what by filter what are the particular biological activity created with our hypothesis. We can make a hypothesis of that particular structure according to the interaction behavior, that is a learning behavior we can implement with the AI model. So we can see that we can create the series of hypotheses by linking a distance between the two fragments, or we can identify the indicators, the important indicators which are used for activity purpose. So like that we can easily make a logical rules our. So if we'll take an example of one rule, we can make a rule: the most important molecules which are being observed in the particular structures may consist of the positive charge, that is the nitrogen. So sp2 hybridized nitrogen is required, and the distance between the A atom and that particular B atom will be the distance between the 5.2 plus minus 5, and from this may be a rule which can be implemented. A second rule, we can implement the requirement of phenyl group. So we can able to see that the phenyl group can be easily be the part of this molecule. So we can say the molecule which consists of phenyl ring will be active. So this type of logical based hypothesis matrix can be done, and according to that we will go with the correlation mechanism. So from that we can able to make our rule with that particular molecules, where suppose whatever number of molecules we have, we can identify that particular molecule, that is a standard molecule. What are we standard training set what we are setting for that particular. So for that whatever molecule we have and the data we have for the interaction, we can make a rule for from that activity. So once we develop that, then we need to go for a checking with the models. So there are a number of screening models. There is one example of zinc model having more than crores of molecules, and we can easily identify with the zinc database molecule. The whatever the model we have developed, the all the skin zinc library we can test with this. And the most common one is the directory of useful decoys, that is DUD, that is available. So for taking an example, if SIR2 protein inhibitor, if you want to process for, that is having a 40 protein targets. So with the use of this protein targets, we can easily identify the interaction of the drugs with the protein target. And from there, we can easily correlate the enrichment curves of different software. So the index software having a 32, 8, 4, and 2, and the random software we can easily this is the enrichment curve which has been given. So it can easily be observed that the software using the AI model has already given the highest enrichment, that is the identification of the known ligand from the percentage data which can be used. So this has clearly given the information about the uh, AI model used in estimation of drug identification of drug. So this interaction factor can also be easily identified, that 1% and 0.15% interaction factor when we compared with the other software, we'll find that this index software is having a what to say highest number of enrichment factors as compared to the other software which are being used. So same, the performance, similarity, and target size can be estimated with the use of this particular software. Now we'll come to the applications part. Now this is the how we can develop a model, or what we can say how we have developed the models, uh, how we need to go for a data, how need to go for a training set, and how we can process for test set. So these four activity we have checked out in this particular AI artificial intelligence. Now how what is the application of this particular in different type of screening. So if we talk about structure based virtual screening, so most commonly SVM based scoring model is being used. So SVM based is a support vector machine based scoring model which is commonly used for small molecular inhibitors. So inhibitor identification, for this purpose we are using the SVM model. And this model is basically in current approach, this model is basically used with the combination of molecular docking, energy minimization, energy mechanism, surface free energy decomposition. So this everything whatever estimation we are using SVM model for structure based virtual screening. And the hit rate, it was observed that 14% hit rate is observed with this particular model. So how you can develop the weight for this particular model? So what you need to do is you need to have a standard drug which is being showing the interaction with the protein, the receptor protein in which it is showing interaction. So once you identify that, you need to make a list of the atoms which are being interacted as a form of weight atom. You need to go with the distance. What are the distance between the atom as a weight distance, the second parameter. You need to go with the charges which are being employed in the interaction as a weight charge, and you need to go with the weight of amino acids which are involved in the interaction, the weights of amino acids. Like this, you need to, these are the weights which you are employed in the model. Once you develop these models, once you develop this everything, you need get the you need to implement this on the AI model. Once you implemented this, according to the next test data, all the calculation will made, and you will get the regression, final regression analysis according to that. And that will be calculated with easily with the formula. W3 is in bracket to W2 R plus B square plus B3. So these are the basic deep models which can be easily used for the developing the architect. Now, the second approach of artificial intelligence is to develop a QSAR model with AI approach. So, the artificial, whatever I have told you previously, is the for virtual screening. This is for QSR model. So when we want to go for a QSR model, we know that QSR model basically based on the mathematical relationship of the molecular property with the that particular quantitative compound. So for that purpose, a best approach is a RF approach. And as we have seen, the previous approach is a SVM approach, which is a support vector machine approach, which is for virtual screening. For docking purpose, we are looking for RF approach, which is known as random forest approach. So random forest approach is the best approach which is used for QSAR based method. And there is a regression model is already been developed for using RF approach, which is known as profile QSAR PQSAR 2.0. So it is being already developed, and new models are still coming in that particular field. So RF model is a very good algorithm which easily enable to identify the chemical relationship between the biological properties and chemical properties of the descriptor analysis. So for that purpose, in this we need to take a database. So we have taken Campbell database, from where we can get a smile code, and from there we need to have a certain experimental evolution such as IC50, EC50, or you can go for T-half, or you can go for permeability. Whatever data you can want to include, you can go with that, and then from there you can go for a descriptive analysis which can be implemented with the training set. Once you develop a training set, then you need to evaluate the individual fragment and its contribution to the activity. Once you develop that, it automatically calculate the huge database and can evaluate the particular compound. So this is an example of development of a anti-hepatitis C agent using the QSAR model. So there is one more important model which is being used in QSAR, that is a Bayesian algorithm model. And this model is again widely used in a number of targets such as kinase target, such as Alzheimer disease, G protein couple receptor, Escherichia coli difolate reductase, amino acid, butyric acid. These are the number of already proteins which have been screened with the use of this particular algorithm. And this apart from this algorithm, ANN algorithm is also a very good component for evaluation of QSAR. So near about all the possible algorithm methods are being used for the QSAR approaches. So here also one more important thing about the QSAR, the property prediction. And all the three methods what I have told you, the support vector machine or random forest or Bayes learning, this all three machines are being used for that purpose. And for repurposing especially purpose, the similarity ensemble approach is being developed for the property predictions. So apart from this, there is one more important role of artificial intelligence is a classification of database. So the huge database can be classified with the use of artificial intelligence. So what to say, this classification helps in improving the virtual screening, as we can get a specific data to use for the particular type of ligands, sorry, the particular type of proteins. So AI helps in identify that, and that will be done with the use of identifying a standard beans. So first the AI, artificial intelligence identify the particular type of structure which are basically required in the form of bean 1, bean 2, bean 3, bean 4, or you can increase that bean also. And according to that, they split out all the database and make a list of the molecule which come under bean 1, bean 2, bean 3, and bean 4. And in the way it can easily classify the data. So this is the particular role of artificial intelligence. Now the artificial intelligence has an important part, that is encoding and decoding. So this is very important for medicinal chemistry point of view, because we are getting data in a small input molecule. So the artificial encoding will convert into the encoder will convert into neural network form, and that neural network form is a representation of this molecule, which will help out in a evolution of property prediction. So once we encode the molecule, go for learning will decode and calculate which type of molecule it is there. So it will help out in estimation of or evolution of the molecule. So when we go for this evolution or interpretation, we can find that we can easily determine the concentration of molecule also. And for determining a concentration of molecule, this is an architect of the molecule. See, this architect include the fingerprint of the molecule, which illuminate the drug concentration. This is the input. This is a fingerprinting, and that estimate the how much concentration of the molecule is. So we need to encode and implement the new database, and according to that it process and results into the output which shows the concentration of molecule. So AI intelligence also help to estimate the concentration of molecule. So this is an example. You can easily see the deep learning. There are a number of parameters which we are fixing: molecular weight, H donor, H acceptor, rotational bond, nitrogen atom, ring, and the TPSA. So these all we implemented, and according to the neural network, they interact with each neural part, and that interaction will finally result into the value of log BB. So this processing itself, by its learning, evaluate the log BB value. So these are the basic tools which are already available in the, you can easily unfold this particular website. You can easily identify these are the software which are available for using artificial intelligence. You can easily go through that, and you can easily employ it for your docking purpose also. So this is all about artificial intelligence. Thank you. Sir, you want still to continue? I have some more things if you want.
Sir,
Yeah.
actually,
So
to
the
the
time
time
is
is
near to completions. I
Yeah.
think you are giving much insight. Basically,